welcome to everybody, and thank you to everybody for attending this webinar organized by the Global Virus Network. Before handing over to Shin-Hee Kim, the Scientific Program Director of GVN, for introducing Dr. Moore, I just want to remind, because some are not necessarily aware of what exactly is GVN about. The GVN, very briefly, is a network which now gathers about 60 research centers worldwide and I believe 12 affiliate centers. And it's really merging the best experts in virology worldwide around activities which merge research, task force, obviously very much on COVID-19 nowadays, but on all human viruses, education and training, and recently with the launching of the GVN Academy, providing mentoring for young students, and postdoc providing fellowships, open call, and we really encourage all of you to have a look at this, and advocacy communication. It's about expertise. And I finally want to draw your attention to the webpage on the GVN website, very much focused on a daily update on COVID-19 variants and vaccines and questions and answers. We are very interested also by your thoughts, because we know that we can improve, although we really believe that this webpage is very useful. So having said that, I hand over to Shin-Hee for introducing Dr. Penny Moore, and thank you very much, Dr. Moore.
Thank you, Dr. Brechot. It is my great honor to introduce today's speaker, Dr. Penny Moore. Dr. Penny Moore is the Center Director for the National Health Laboratory Service, Johannesburg, National Institute for Communicable Diseases, South Africa. Dr. Moore is the South African Research Chair of Virus Host Dynamics and the Reader, Associate Professor at the University of the Witwatersrand and the National Institute for the Communicable Diseases. She is also Senior Honorary Scientist, Center for the AIDS Program of Research, University of Kwazulu-Natal, and the Adjunct Member of the Institute of the Infectious Disease and Molecular Medicine, University of Cape Town. She received her bachelor and the master degree from the University of Witwatersrand, South Africa, and her PhD from Royal Free and University College Medical School, UK. In the past 15 years, Dr. Moore has worked on the HIV. She has published more than 100 papers focusing on HIV neutralizing antibodies and their interplay with the evolving virus and the vaccine development. Her research is currently funded by the NIH, the South Africa Medical Research Council, and the International AIDS Vaccine Initiative, and the South Africa National Research Foundation. With the COVID-19 pandemic, she has extended her research to SARS-CoV-2. This year has been already productive year to her, with a variant emerged from South Africa. Her laboratory showed that the variant escaped neutralization by South Africa COVID-19 donor plasmid and induced cross-reactive neutralizing antibody. Dr. Moore also participated in evaluating safety and efficacy of the AstraZeneca vaccine against the variant in South Africa. So we'll hear more about her research progress today. And Dr. Moore, thank you for taking your time and giving your talk today. Thank you.
Thanks, Shin-Hee. Let me see if I can share my slides. Are they up? Hope so. Hello, everyone. And thank you for the opportunity to speak to you today. So yes, I'm going to whip you through some of the work that we've been doing over the last year, looking at SARS-CoV-2 in South Africa, with a real emphasis, as you've already heard, on the variant called 51YV2 or B.1.351, which was first detected in South Africa and characterizing the immune response to that virus. So 501YV2 was really detected during the emergence of the second wave in South Africa. We've been through 2 waves and are probably about to head into a 3rd wave, we think. The numbers are slowly increasing. The second wave happened somewhere around, well, I guess towards the end of late last year. And as the increasing numbers in South Africa started spiking, that resulted in an intensified genomic surveillance across South Africa, particularly looking in areas where the wave started to emerge. And that genomics team, which I'll tell you a little bit about on the next slide, led by Tulia D'Oliveira, were very quick to detect a new variant that had emerged in South Africa called 501YV2. If you look at the panel here in the middle, you can see the emergence of that new variant in yellow. You can see that it was detected as early as October of last year, and by November or December had really started dominating all the sequences that were being generated across South Africa. And that was rapidly followed by the spread of 501YV2 from the Eastern Cape and really across South Africa. So the ability of South Africa to pick up this variant very early was largely testament to the Network for Genomic Surveillance in South Africa, or NGS-SA, which is a really incredible network of viral sequences across South Africa, led, as I mentioned, by Tulia D'Oliveira. But it's a team that works closely with multiple sites, largely built on the existing laboratories that were already in place across South Africa and that form part of our National Health Laboratory Services. So it leveraged existing infrastructure and also leveraged skills that had been generated for viral sequencing analyses specifically, many of which came out of HIV studies that had been ongoing in South Africa for a very long time. And you can see here in the slide some of those sites at CRISP, or the University of KwaZulu-Natal, the University of the Free State in Bloemfontein, 2 sites at Groote Schuur Hospital and Tygerberg in the Cape, and then where I'm based here in Johannesburg at the National Institute for Communicable Diseases. But despite the fact that the media continue, unfortunately, to refer to this as the South African variant, 501YV2 is certainly not limited to South Africa and has now been detected in many countries across the world, more than 30 last I checked, and this is from the 5th of March from a very reliable source called Wikipedia. So it's certainly started to spread across the world, in some cases showing community transmission. So I thought I'd start by reminding you of what defines this SARS-CoV-2 variant 501YV2. As with all the other variants that have been described, there are many mutations across the genome, as you can see at the top here. But if you zoom in and look in more detail, the mutations that we were particularly concerned about are present in the spike protein. And the reason for that is obvious. The spike protein is really the major target of the immune response, particularly from a vaccine perspective. And so mutations that are present in this region are always concerning from an immune escape perspective. If you look across here, you can see that there are several mutations that define 501YV2. The variant does include the D614G mutation that's common to almost all global viruses now. Also, 3 mutations in the receptor binding domain, a series of mutations in the N-terminal domain. And the reason that we were particularly concerned by the location of these mutations is really based on the fact that the neutralizing antibody response to SARS-CoV-2 infection is almost entirely dominated by antibodies to the receptor binding domain and the N-terminal domain, both of which happen to be where these mutations exist. So this on the left-hand side here shows you some examples of this. These are monoclonal antibodies, class 1 and class 2 monoclonal antibodies. These classes were defined by Christopher Barnes and Pamela Bjorkman and define kind of antigenic supersites. And you can see that, that class 1 and class 2 antibodies, as have been shown in many, many monoclonal antibody isolation studies, are really responsible for a lot of the neutralizing antibody response to SARS-CoV-2. The N-terminal domain, although initially thought to be less frequently targeted, I think is becoming increasingly implicated as a really dominant target for neutralizing antibodies to SARS-CoV-2. And that's an example of that would be 4A8, but since then many other monoclonal antibodies have been isolated to this target. So we, as you heard from the very kind introduction, my group have been focused on HIV for a very long time. And we've been interested in understanding the neutralizing antibody response to HIV infection and vaccination. And for that had used a very well-described neutralizing antibody assay, that's a pseudotype virus assay. And so when we started to move into the SARS-CoV-2 space, we essentially repurposed what was an existing assay to use to adapt it towards the spike protein of SARS-CoV-2. And really the workhorses of this have been 3 spike proteins, which we then use to pseudotype a lentiviral HIV-based backbone to make pseudovirus particles. We've really focused on comparing the wild type D614G, which was the variant that was circulating in South Africa before the emergence of 501YV2, the full 501YV2 variant at the bottom here in black, and then an RBD triple mutant that contains only these mutations at positions 501, a mutation I'm sure you're very familiar with, also E484 or EEK, and then the K417N mutation. And we focused on these given the predominance of RBD responses during infection and in vaccination. And we use a cell line that was developed by Mark Farzan, a 293T based cell line that overexpresses ACE2. That has been a very useful platform for us to be able to really rapidly generate neutralization data, both in infection and in vaccination. So I'm really going to tell you 2 separate parts of the story today. And the first is work that was really led by Kurt Wibmer and was published earlier this year in Nature Medicine, which showed that SARS-CoV-2 501YV2 escapes neutralization by South African COVID-19 plasma. And this was work that was also heavily involved Dr. Janelle Beeman, who's shown in the picture below, but many other people in our lab. It was a huge effort over as the variant emerged over the Christmas break. So I was very popular with the lab. I'm going to show you first data looking at monoclonal antibodies. So we hypothesized that SARS-CoV-2 501YV2 would be resistant to monoclonal antibodies because of the location of the mutations. This is looking first at these class 1 monoclonal antibodies to the RBD domain, the receptor binding domain. These antibodies are typically highly dependent on position 4 and 7. And as you remember, K417N is one of the mutations that defines this lineage. And indeed, when we took 3 examples of this class of antibodies shown here at the top, and we tested them both in ELISA and in neutralization assays against the wild type original variant in black and 501YV2 in red, both in, here's the ELISA at the top, neutralization at the bottom, and the maroon curve shows you that viral spike that has just the 3 mutations within the RBD. And you can see that for all 3 of these monoclonal antibodies, we saw complete knockout of binding and also of neutralization. We saw a very similar thing with the second class of RBD-directed monoclonal antibodies. Class 2 antibodies are heavily dependent on this position 484, which again had a charge change, E484K mutation. And again, we saw the same thing here, again, 3 different members of this class of antibody resistant to 501YV2 neutralization, both as measured by ELISA and by neutralization. And then lastly, looking at the monoclonal antibodies to the N-terminal domain, again, good reason to think that this variant would escape these monoclonal antibodies because many of them are dependent on position 246 and on the N5 loop supersite, which is where in 501YV2 there's a deletion. So those 2 changes amongst many of the other changes in the NTD essentially result in a sort of a remodeling of the N-terminal domain antigenic supersite. And that confers resistance to this monoclonal antibody, which is 4A8, again, both in ELISA at the top and neutralization in the bottom. So overall, this data convinced us that the 501YV2 lineage had escaped from 3 classes of therapeutically relevant monoclonal antibodies. Since then, there have been several antibodies that have been described in the literature that do show some activity against 501YV2. But I think important to point out that many of these are the antibodies that have already got emergency use authorization and that do take a significant hit against this variant. The next thing that Kurt did was to ask the same question for convalescent donor plasma. And to do this, we used a cohort of 44 individuals who'd previously been infected with the original D614G variant. These were people who were recruited during the first wave of infection in South Africa before the emergence of the variant. And what Kurt did was to select a range of titers. Some of these up in the high 10,000 range. These were people who were hospitalized. And then a range of more moderately ill people with more moderate neutralization titers against the original variant. But as you can see here, when Kurt tested these against 501YB2 in the pseudotype neutralization assay, there was a 9-fold reduction in neutralization. And more dramatically, when you put that into a pie chart, what that meant was that 48% of these sera no longer showed any activity against 501YB2 in our assay. And of course, that has significant implications both for immune escape and reinfection, and that's something that we can discuss, but also for vaccine efficacy in South Africa. I think the thing that somehow often gets lost in this particular set of data is the fact that although we saw a really substantial knockout in neutralization, that knockout, when you look at binding antibodies, occurs much less effectively. And that data shown here, this is area under the curve data with ELISAs using an RBD plus SBD1 protein. And you can see that there is binding against the original variant, the D614G on the far left. But there's also quite high preservation of that binding against 501YB2. So although there's a fold reduction that ranges anywhere from sort of virtually nothing all the way to, I think the highest is 16-fold, you do see maintained binding against the variant in polyclonal plasma responses. So it escapes neutralization very effectively, binding antibodies less so. So I mentioned that our concern was the impact of 501YB2 on vaccine efficacy. And of course, this is something that many people have been interested across the world. And I'm showing you a subset here of published data, looking at the Moderna and the Pfizer vaccines. If you look at the far left and the middle panels, D614G, or the original variant, is shown in black. The pink is the UK variant, which I'm not really going to focus on. But in orange is the South African variant. And you can see that when people tested vaccinee plasma, either from the Moderna or the Pfizer trials, there was a really substantial loss of activity in vaccinee plasma, anywhere between 6.5 and 8.5-fold in this context. So certainly, as with convalescent plasma, vaccinee plasma are impacted in their ability to neutralize this variant. We focused particularly on the AstraZeneca Oxford vaccine. And the reason we focused on that vaccine was that many of you may know that South Africa has a very slow, unfortunately, very, very slow vaccine rollout. At the time, the only vaccine that was present in South Africa and had been tested in South Africa was the AstraZeneca vaccine. And so amidst great fanfare, that was the vaccine that was being imported into South Africa for a rollout. At virtually the same time that airplane bearing these doses of the AstraZeneca vaccine arrived in South Africa, we showed in using the same laboratory assay that the 501YB2 variant was essentially completely resistant to sera from people who had received the ChAdOx or AstraZeneca vaccine as part of a phase 1/2 trial that had happened in parallel to the emergence of the variant in South Africa. So essentially, this is a trial that had not been set up to evaluate efficacy against 501YB2. It had been rolled out literally at exactly the same time that 501YB2 emerged, and so coincidentally ended up being an experiment that did measure the efficacy of the AstraZeneca regime against 501YB2. I'll show you first the laboratory data, which is here. It's the same format as the data I showed you before. So here you have a series of vaccinee plasma with titers ranging from less than 100 to more than, I think, 1 in 2,000, 2,500. And you can see when we tested them against 501YB2, 84% of these vaccinee plasma showed no detectable neutralization of the 501YB2 variant. So that was concerning. At the same time, even more concerning was the results of the clinical trial that I mentioned. This was a clinical trial that was led by Professor Shabir Mahdi. It was a relatively small trial, and it was not powered to detect protection against severe disease. But this 2-dose regimen of the AstraZeneca vaccine in South Africa showed no protection, no detectable protection at all, really, against mild to moderate COVID-19 that was due to 501YB2. And you can see the Kaplan-Meier here, the placebo in pink and the vaccine in blue, and really no significant difference. But as I mentioned, it was not a vaccine that was a vaccine trial that was geared to test protection from severe disease. And so we don't know in this trial whether the vaccine had any impact on severe disease. And I mention that because as with studies of convalescent plasma, when we looked at binding antibodies, again, we saw a much less dramatic drop-off in activity against 501YB2. So I think it's not impossible that this vaccine, in fact, I think it's very likely that this vaccine would in fact protect against severe disease, even against SARS-CoV-2, 501YB2, but we simply don't have the data to show that. And I think we can discuss this in the question and answer time, but it resulted in a lot of argument within the South African scientific and ethical community about whether that AstraZeneca vaccine should still be rolled out, at least in a subset of South Africans, given that at the time no other vaccine was available. This is unpublished data that asks the question about the Johnson & Johnson or Janssen vaccine. This is a vaccine that is currently it's not being rolled out because until very recently didn't have approval, but it was being tested essentially in a phase 3B implementation trial, which was a way in which the country was able to start to vaccinate healthcare workers. As I'm sure everybody knows, that vaccine trial has been halted in South Africa as a consequence of the FDA decision to halt the US Johnson & Johnson vaccine, though I imagine that this will be restarted again very shortly in South Africa. But we again ask the same question here. You're probably getting very used to seeing this formatted data, but again, taking a range of sera from vaccinees, testing them against the original variant and the 501YB2 variant. And again, in fact, almost identical to what we'd seen with the AstraZeneca regime, we saw 84% knockout. But here, of course, the major difference is that in South Africa, we've had a phase 3 clinical trial, the Ensemble trial, of this vaccine in South Africa against 501YB2. It was a vaccine that was conducted at almost exactly the same time as the AstraZeneca trial, a much larger trial than the AstraZeneca trial. And here we saw 64% protection against moderate to severe disease and 82% protection against severe or critical disease, despite the fact that, as you can see, we see no detectable or very little detectable neutralization against 501YB2 in our assays. And I think this is an interesting example of the potential limitations of laboratory data, though they're certainly informative, in using these as a simple measure of whether a vaccine will work or not. It's clear that there are other immunological players here that confer protection from critical disease in the Johnson & Johnson vaccine. And this essentially confirms exactly what we have shown you before, is that despite the loss of neutralization, the binding antibodies are retained against 501YB2 in people who are vaccinated with the Johnson & Johnson vaccine, perhaps providing a mechanism for that protection from severe disease that we know exists with this vaccine. So this is the first two-thirds of my talk, which I think shows fairly comprehensively that 501YB2 escapes 3 classes of therapeutically relevant monoclonal antibodies, escapes neutralizing but not binding antibodies in convalescent plasma, and also neutralizing antibodies but not binding antibodies in sera from 2 vaccine regimens. So now I'm going to, in the last part of my talk, change gears and talk to you about a more recent study that came out a couple of weeks ago in NEJM that was led by Thandeka Gwetemoya and Mshudu Matsilindile in our lab, and their photos are shown here, which asked the question of how well recognized 501YB2 is by the immune system. So there were really 2 questions. Given the presence of these escape mutations within particularly the RBD and NTD of 501YB2, would that somehow compromise the immunogenicity of 501YB2 during infection, or would it elicit an equally robust neutralizing antibody response? And then the second question, which is there cross-reactivity in either the binding or the neutralization of antibodies that are triggered by 501YB2 infection in South African donors? We addressed this in a cohort of 89 hospitalized patients who were recruited in January and February of this year in the Groote Schuur Hospital. And this is a picture of happier days, actually, at the Groote Schuur Hospital. And the University of Cape Town is where this Groote Schuur Hospital is based, is currently on fire. And so it's much of the bottom of Table Mountain. And so it's a very distressing time, I think, for Cape Townians who are watching the library and the res and much of the university campus being burned at the moment. But before that happened, we collaborated with a wonderful group of people, Ntobeko Ntuzi and Wendy Burgers and Carolyn Williamson, Wendy and Carolyn being very long-term HIV collaborators of ours at the University of Cape Town, Tulio at CRISP, our team at NICD, and then we worked with Teresa Rousseau and Michael Boswell, who also had a cohort of hospitalized donors who they had recruited during the first wave of infection. So essentially what that did was enable us to compare immune responses to the original variant in the first wave of infection with this cohort of hospitalized patients infected in the second wave. At the time, in January and February, 501YV2 had already become the dominant strain in South Africa. That's shown on this timeline, vertical timeline here. So blue being the original variant, and again, yellow being 501YV2. And you can see this is data from the Western Cape, where Cape Town is based. And you can see that almost all infections for quite some time already in this region had been through 501YV2 rather than the original variant. And we were able to confirm that in a subset of nearly 30 of the 89 infected individuals that we studied. We only had sequences available, only swabs had only been collected for this subset, or had only been stored for long enough for the subset that we were able to sequence them. This is a phylogenetic tree showing you global sequences. In yellow are the 501YV2 sequences from South Africa, and the green dots here at the bottom are the individuals that we studied as part of the Groote Schuur cohort. And you can see that all of the 28, if I recall rightly, all of the 28 sequences that we were able to obtain clustered with 501YV2, confirming that this was likely the source of all 89 infections in our cohort. We started by asking about binding antibodies. On the left-hand side here are binding antibodies to the full spike, and on the right-hand side to the RBD plus a little bit extra. And this you can see the vertical axis in both cases 501YV2, spike mutated to match that variant, and along the bottom is the original variant. And you can see that there's a slight reduction in activity against when tested against the original variant. In other words, there's preferential binding to the 501YV2 spike in these individuals who were infected with 501YV2, but there's substantial cross-reactivity as we've seen in all the other cohorts we've looked at. The next question we asked was whether the neutralizing antibody response to 501YV2 was equivalent to what we were used to seeing in people infected with the original variant in South Africa. And so we compared the Steve Biko cohort from the first wave and the Groote Schuur cohort from the second wave. 501YV2 is the Groote Schuur cohort, and the original variant, these are hospitalized individuals infected with the original variant. And you can see that there's not really a marked difference between these two sets of neutralizing antibody titers. It's difficult to say more than that there's not a marked difference, because these were hospitalized individuals, and at that point in infection, as I'm sure you all know, the immune response is really ramping up at a very rapid rate. So these cohorts, it's very difficult to match hospitalized cohorts to the exact day from diagnosis or from better infection. But I think all we'd be comfortable in saying, because these two cohorts probably do differ by a couple of days, is that there's certainly a potent neutralizing antibody response to 501YV2. And then the last question we asked was about the cross-reactivity. So this is the data that I showed you on one of my very first slides. This is Kurt's data looking at the original cohort of 44 individuals who were infected with the original variant, and showing that they showed really dramatically reduced titers against 501YV2. What we did next was to ask the opposite question, was to take the people who've been infected with 501YV2 and ask how effectively the antibodies that they had were able to neutralize the original variant. And that data is shown here. This is a subset of 57 of the 89 individuals for whom we had neutralization data. But you can see that there's a much less dramatic drop-off in neutralization, as opposed to a 9-fold reduction that we saw in the initial cohort. We only see a 3-fold reduction in titers here. and only 7% of these sera are no longer able to neutralize the original virus. So there does seem to be much greater levels of cross-reactivity in the immune response, in the neutralizing antibody response to be specific, triggered by 501YB2 compared to the immune response that was triggered by the original variant. It's not only something that's specific to the original variant, it's also an intriguing set of data here for a subset of only 10 individuals that asks what happens when we test sera from people who are infected with 501YB2 against 501YB3 or P1, the variant that was first described in Brazil. And you can see here that actually there's surprisingly a slight increase in neutralization of 501YB3, and certainly in 10 of 10 sera being able to very effectively neutralize that variant. And this is something that we're expanding on at the moment. So the 501YB2 variant certainly elicits robust binding and neutralizing antibody responses. Sera from 501YB2 infections show cross-reactivity, neutralizing both their own variant, the original variant, and 501YB3. And I think that that's encouraging, particularly as many of the vaccine manufacturers have already moved and have done so in many cases extremely fast to incorporate the spike from 501YB2 into their vaccine platforms. Certainly Pfizer and Moderna have already done this and moved forward. In fact, are beginning immunogenicity testing at the moment. And I think it does suggest the possibility that there is something intrinsic to the spike from 501YB2 that may somehow trigger more cross-reactive neutralizing antibodies. And if that's true, then it may turn out that vaccines that are based on the 501YB2 spike may be a promising step in the right direction in terms of vaccine development, especially as we moved on to try to deal with continually emerging variants. And so I'll stop there. The lab has been absolutely incredible. They've moved extremely quickly to implement a whole new set of assays to learn about SARS-CoV-2, as the rest of the world been doing really very quickly. They worked incredibly long hours over Christmas and have done ever since to really react, to understand what's happening to SARS-CoV-2 epidemic in South Africa and the implications of what we're seeing in South Africa for the rest of the world. So they've been absolutely incredible, and if any of them are listening now, that's my thank you. But yeah, this is our team over here. You can see it's a large group, and I'm very grateful to work with them. Our collaborators at the University of Pretoria, the University of Cape Town, the University of Kwazulu-Natal, and Wits University. We've been talking a lot within the country with one another about what the key questions are that we need to address nationally. So we have a 501YB2 consortium that gets together once a week, and it's been led largely by Willem Hanekom and Tulia D'Oliveira, and has been really incredibly informative and energizing and gearing South Africans to work together in ways we haven't, I don't think we could ever imagine. The clinical teams and the participants without whom we couldn't do any of this work, and our funders. And I'll stop there and I'm happy to take questions. Thanks.
Thank you, Dr. Moore. It was really nice presentation, well organized, and really updated information for everybody. So we'll take the question and answer. So can you say your question and answer?
Yes.
Okay.
Yes, I can. Would you like me to read them out, Shani?
It's up to you.
Okay. Okay, so the first question is, do you think the mRNA vaccines will confer sufficient protection for the moment, even against 501YB2, and then maybe boost with a modified vaccine in 2022? So there is quite a lot of neutralization data for the Pfizer and the Moderna vaccines. In general, whether we look at infection or vaccination, it doesn't really matter which sera we look at. There's a titer dependent loss. So if you start high, you tend to retain some activity against 501YB2, whereas if you start low, you show no detectable neutralization. The mRNA platforms have the advantage of generating really incredibly high titers. So in most cases, there is still, in all the lab assays that have been conducted, some residual neutralization of 501YB2, which is very reassuring. We also have efficacy data from a small subset of people, about 800 only, of Pfizer vaccinees in South Africa who did show good protection. There were very good indications that that vaccine would protect against 501YB2. So I think it's very likely that there will still be some degree of preservation of activity against 501YB2 and against the other variants that share many of the same mutations. And then yes, maybe boost with a modified vaccine. I think that's something that we're going to have to look at very carefully, and it's going to be almost entirely dependent on what comes next in the way of variants. So I think that's the only question I see at the moment. Ah, there's another.
It's the one more question.
Do data suggest whether selective pressure from variants came from improved transmissibility or immune escape? Yeah, so the 501YB2, as I mentioned, does include the 501 mutation that is associated with increased transmissibility, increased binding affinity for the ACE2 receptor. So there may be that. We actually don't have firm laboratory data for 501YB2 that I'm aware of that shows increased transmissibility, but certainly it was associated with a massive increase in numbers in South Africa. And I do think it's very likely that that there is increased transmissibility. Unfortunately, in South Africa, the second wave made this slightly difficult to disentangle because the health systems were under such pressure that it's also possible that we saw increased mortality because of overburdening of the healthcare services rather than the virus. So it's very difficult to disentangle those two things. However, my guess is that this is largely determined by immune escape. So there've been a series of sera surveys that have been conducted in South Africa over the last year, and there's an incredibly high burden of previous infection in South Africa. In some of the surveys that have been done, for example, around the Baragwanath Hospital, Chris Hani Baragwanath Hospital in Soweto, which is a very high density urban area, the people, Shabir Mahdi, who did that study, saw in excess of 40% sera positivity in that cohort. So I think given that backdrop of huge immune pressure on the virus at the population level, in places like South Africa and in Brazil, and in many other countries where there's a huge burden of infection, and where people are certainly mounting a very robust immune response, and it is very targeted to the RBD and the NTD, I think the likelihood is that immune escape is a major driver for selecting these variants. Yeah. Damien, hello. Have there been any new isolates that have attained an additional large number of mutations on top of 501YB2? Yeah, it's a good question, yeah. So within 501YB2, there's already kind of a suite of mutations that exist in different constellations, I think is the word people tend to use. But to my knowledge, while we're starting to see spotty mutations come up, I don't think there's been an entirely new burst sublineage within 501YB2. I'm sure it'll come. Mark, in measles, there's a syndrome where incomplete neutralization results in allergic reaction to the virus when children get wild type. Is there any possibility that those immunized to Pfizer would get allergic reactions when infected with the South African variant? We don't know. I don't know, maybe other people know, but I don't know the answer to that. I know there was concern about antibody-dependent enhancement, has been for a long time in the SARS-CoV-2 field, but I think that's a slightly different thing to what you're referring to. I think it's entirely possible that we're going to see high levels of reinfection because of immune escape. And so whether you're talking about a vaccination background or an infection background, I think we are going to be looking at many cases where people are infected and then are reinfected. We've been seeing reinfections in South Africa from the very beginning. I think there's probably a greater level of reinfection now with the emergence of 501YB2. I haven't heard any description of allergic reactions, and I don't know the syndrome. But I wouldn't rule out the possibility that we will start seeing switching of the immune response to sort of an original antigenic sin, and I don't think we know yet how that will pan out. Bob Geller, I arrived late, but can you say how the COVID-19 situation in South Africa is evolving? Yeah, we had for those of you who don't know, the first wave of SARS-CoV-2 infection in South Africa happened during a really draconian, incredible lockdown, where people weren't allowed to leave their homes, weren't allowed to do anything, weren't allowed to walk their dogs. And so we had a lockdown like that. I think slowed down the first wave dramatically and gave the medical folks time to at least build some sort of facilities. The second wave was very fast and came very hard, but resolved with much the same kind of kinetics as the first wave. At the moment, our situation luckily is very much under control. We have less than, I think, 4% positivity in our testing rates at the moment. And the numbers are very, very low, but they are starting to creep up. And we certainly are concerned that, having had the recent Easter holiday, Easter is a big deal in South Africa, a lot of people travel. So we're watching anxiously for the third wave. Jose, that's a nice name to see. Do you see a role for binding non-neutralizing antibodies in protection? Yeah. One of the postdocs in our lab, Simone Richardson, has set up with her team a suite of Fc effector functions. And she's busy looking at those both in the context of immediately in convalescent plasma, comparing hospitalized, non-hospitalized, HIV positive, HIV negative. And she's seeing some interesting things. That team is now extending that to vaccination. So certainly there's a robust Fc effector function going on. And I think there's data from several other studies that suggest that Fc effector function may contribute both to protection and to resolution of illness in SARS-CoV-2, yeah. Can you comment on the cellular response is in the panelist chat. Yeah, so a very close collaborator of ours, Dr. Wendy Burgess, is working to address the question of whether T-cell responses are impacted by the 501YV2 variant. Certainly, many of the mutations in 501YV2 do exist within cellular epitopes, so I think there's a good reason to think that they may be impacted. I know there's some data from the SETI lab that shows that the impact may not be as dramatic as it is for neutralization, but I think that data will be coming out in the next. Generally, I'm aware of several studies looking at this at the moment, and I think we'll see data about this in the next couple of weeks, I would say, coming up on bioRxiv. How is South Africa monitoring reinfections, and what's your opinion on their importance? Yeah, so reinfections are difficult to monitor, particularly in the context of high levels of seropositivity, because many of those reinfections that may be going on are probably not being detected. So, you know, if 90% of infections are asymptomatic, and then the next 90% of infections on top of those are also asymptomatic, then we're probably missing many of those reinfections. There is an active attempt to identify reinfections. So anybody who is found to be PCR positive more than 90 days after primary infection is regarded as being a reinfection. And many of us are working to try to understand the immune response to that and studying the breakthrough second virus and trying to understand whether that is 501YV2 or not. So at the moment, there are kind of sporadic case reports of this, and there are more and more of those happening. But I think it's difficult to quantify. And that's me, I think.
Yeah, I think we received all the questions this morning. So thank you for your time and giving really nice presentation today.
Thank you.
Thank you so much. Very good presentation.
Thank you very much. Thanks for having me.